My name is Jeremy Guillon, and I'm a PhD student at the Aramis Lab inside the Brain and Spine Institute located in Paris. I'm working under the supervision of Fabrizio De Vicofalani, and today I'm going to talk to you about the loss of a new type of hubs called the interfrequency hubs in Alzheimer's disease. Let me first give you a brief introduction of what Alzheimer's disease is. So I will give you one number. There is one person every 4 seconds in the world that is diagnosed as Alzheimer's disease. So it is by far the primary cause of dementia. The main symptoms are the progressive loss of short-term memory, the loss of motor skills, language skills, and at later stages of the disease, disorientation and confusion may appear. It is a neurodegenerative disease, meaning that progressively your neurons will lose their structure and or function. It will cause the brain to shrink and the ventricles to enlarge, as you can see on the picture here. Now the question is, how do these structural damage affect the functional connectivity networks? So to answer that question, what we did was to extract at each frequency band a functional connectivity network. Why? Because it is known that frequency bands are associated to specific function. You would have, for instance, the alpha bands associated to attention or the delta band associated to emotion. We used the spectral coherence, which is a kind of cross-correlation but in the frequency domain on source-projected MEG signals. MEG standing for magnetoencephalography. Now, instead of analyzing all those functional connectivity networks independently, as it is done usually, we combine them in a specific topology called a multiplex, where similar nodes in all the layers are all linked together. It allowed us to define this new type of hub called interfrequency hub as nodes who have that have a high multiparticipation coefficient. A multiparticipation coefficient is a measure that gives you that tells you how evenly connected a node is across the layers. For instance, on this example on the right-hand side of the slide, you have the node A that could be considered as an interfrequency hub because it has the exact same number of connections in all the layers, leading to an MPC value of 1, the maximum. On the other hand, you would have the node B in blue that could not be considered as an interfrequency hub since it has all its connections concentrated in only one layer, leading to an MPC value of 0, the minimum. We applied this methodology to 25 multi-frequency functional connectivity network of healthy subjects, and we realized that first, the biggest interfrequency hub that is on the red cingulate area is also known to be structural hub in the literature. We also realized that the MPC values are almost always high, meaning that brain regions manage to distribute their connections across evenly across different frequency bands. And this biggest hub also appeared to be significantly different degree distribution in Alzheimer's disease. On the right-hand side of the slide, you have the degree distribution across all the layers of that specific region. And the red bars correspond to the ones of Alzheimer's disease patients. And as you can see, on high frequency bands, there is a loss of degree proportion, whereas on lower frequency band, you have an increase, an abnormally increase of functional degree. I'd like to thank my amazing team and you for your attention.